Hello everyone, welcome to MCAT Max, and today we're going to be looking at a passage from the bio/biochemistry section of the MCAT. Uh, these questions are from Jack Weston, and they're pretty good for practice, but let's walk through them and see how we can tackle them. All right. Let's see. The first sentence says that myogenic regulatory factors MyoD1 and myogenin act in concert with epigenetic mechanisms to control skeletal muscle differentiation. So when you're reading MCAT passages, what you want to know is you want to pay attention to the very first sentence, because often the first sentence will give you the main topic of the passage, and you can kind of draw on knowledge that you know about the topic. For example, if you know about skeletal muscle, since this passage is going to be about skeletal muscle, you want to keep that information in mind. And the main subject is so a lot of these times the biology passages, there are going to be a ton of molecules mentioned, like transcription factors, genes, just a ton of subjects mentioned. So you want to keep track of them in your head and really identify what's going on, because it's going to make the questions a lot easier to answer. Okay. So here it says there's myogenic regulatory factors, and these are MyoD1 and myogenin, okay? And it controls skeletal muscle differentiation, okay? So it tells us the MyoD1 is expressed in undifferentiated proliferating myoblasts, okay? That's something to keep in mind. And these are the embryonic precursors of myocytes, okay? So its primary functions are to commit myoblasts to the skeletal muscle lineage and induce cell cycle arrest. Okay, so what does that kind of mean? So what are we talking about? We're talking about a regulatory factor. A regulatory factor affects the fate of cells. So it tells us that MyoD1 is expressed in undifferentiated myoblasts. So let's kind of draw out what that means. So let's say we got a cell here. Okay, let's say this is a myoblast. It's going to have DNA here inside the cell. And that DNA is going to code for MyoD1, right? Because it says MyoD1 is expressed in the myoblast, okay? But what MyoD1 is going to do is MyoD1 MyoD1 is going to act on the cell. MyoD1 is going to act on the cell to cause differentiation into a new cell called a myocyte. So the cell is going to differentiate, and it's going to be in the G0, the quiescent phase, and it's not, and cell cycle is going to stop, right? Because it's going to be differentiated. So cell cycle stops, right? The cell cycle is going to stop because the myoblast will differentiate into a myocyte. So it's going to stop dividing. So these cells are undifferentiated cells, the myoblasts. They keep dividing, but they don't differentiate. So these cells are going to express the MyoD1, our regulatory factor. This regulatory factor is going to cause the cell in some fashion to differentiate into a myocyte. The myocyte is not going to divide anymore. It's just going to be differentiated. So oftentimes you will have cells that just divide. But then at a certain point, the they will differentiate and exit the cell cycle. So the cell cycle, you have G1, S, G2, M, but, so, you can exit the cell cycle when you differentiate and go into a quiescent state. Quiescent state. Quiescent state. Where you no longer divide because you are differentiated. Okay? Alright, so that is what MyoD1 does. Okay? And feel free to pause the video anytime if you want to go back, but I'm going to erase for now. Okay. All right. So we talked about MyoD1, right? Well, let's talk about myogenin because it talks about both of them here. It says myogenin is responsible for the fusion of myogenic precursor cells to either new or previously existing fibers. So what do we know about muscle fibers? We know muscle fibers are made up of long fibers. Oops, that was a bad drawing, but okay. They're made up of long fibers with multiple nuclei in it. Okay, let's say this is our nuclei. So those nuclei, these long muscle fibers have multiple nuclei. They're fused together. So originally, this came from a bunch of cells with nuclei in them, and they all join together. And that is through myogenin. So it causes the precursors of the skeletal muscle cells to fuse together to give the eventual skeletal muscle cells to the muscle fibers. So we know muscle fibers are multinucleated, and they're derived from precursor cells, which each have their own nuclei, which all combine together when the cells fuse, and that's through myogenin. Okay. So here it says MyoD1 hetero. So we have MyoD1 heterodimerizes the E protein 12. So let's try that out. So MyoD1. Let's say this is the monomer MyoD1. It's going to heterodimerize, which means we're going to have a complex of 2 units, and each unit's going to be different. So MyoD1 and E protein 12, they are going to associate. Okay? They're going to associate. And once they associate, this heterodimer, it's a heterodimer because MyoD1 and E protein 12 are each different parts of the dimer. Like the dimer is not symmetrical, it's not the same. They're going to bind protein X. So let's add protein X. Okay. So now we have our complex. So guys, like when they mention to you a bunch of like proteins and they're talking about how they're interacting with one another, it can be really easy to get confused about like the interactions and stuff like that. So it might be helpful on scratch paper to like draw it out, just like a quick little sketch to like help you stay organized mentally throughout this whole passage. Okay. And what it tells us is that positive residues within the protein X E-box binding domain are able to bind E-box DNA sequences in the promoters or skeletal muscle specific genes. Okay, so let's kind of draw out what that means. So we have DNA right here. Okay, let's call this DNA. So downstream, these are going to be our skeletal muscle genes. This protein complex that we were talking about earlier is going to be able to bind to the promoter of these skeletal muscle genes. So it's going to control the expression of the skeletal muscle genes. Okay? And it binds specifically to the E-box DNA sequences of the promoter. So remember that it said that MyoD1 controls the commitment of myoblasts to the skeletal muscle lineage. So this is how it's doing it. It associates with the E protein, forms the heterodimer, then it associates with protein X. This complex then binds to the promoter of the skeletal muscle genes and increases its expression through binding to the E-box DNA sequences and the promoter. And it increases the expression of the skeletal muscle genes so that these myoblast cells can differentiate into the myocytes. So that's its method of action. Okay? All right. So now that we've done that, it tells us that MyoD1 expression is increased by P21 clip. So we know P21 clip is going to increase the amount of skeletal muscle, because since it increases the MyoD1, you're going to have more of this protein complex and more activation of the expression of the skeletal muscle genes. So you're going to have more differentiation into skeletal muscle. So let's draw a plus sign next to P21 clip. Okay. On the other hand, inhibitors of DNA binding proteins are able to sequester E proteins, preventing heterodimerization. Okay, so if you have an ID protein, it is going to decrease the E proteins. So you will not have, because it's gonna sequester. Sequester means to isolate. So it's gonna isolate this E protein, and you are going to decrease this protein complex. If you decrease this protein complex, it cannot bind to the E-box DNA sequences within the promoter. So that is going to decrease skeletal muscle differentiation. So that's why it is going to minus sign. It's going to decrease skeletal muscle. Okay? So we talked about the ID proteins. It says here MyoR can bind to and inactivate the E-box binding domain of protein X. So if you inactivate, so let's say this MyoR, what do you think it's going to do? Is it going to decrease or increase skeletal muscle? So it's going to inactivate protein X's binding domain of the DNA sequences. So if you inactivate the DNA binding domain of protein X, this complex is not going to be able to bind to the DNA sequences. It will not increase differentiation of the skeletal muscle, and you will have less skeletal muscles. So it's going to decrease decrease skeletal muscle. So, okay. So now we've gotten a good idea of like what all the relevant proteins are. What are like the contents of the passage, like what's going on in the passage. We've gotten a good idea of that. And whenever you do an MCAT passage, you want to have a good idea of what's going on, because when it gets to the questions, if you have a good organization in your head of what's going on, it's going to make the questions way easier to answer, especially in these biology passages where there's so many components. So let's move on. Okay. And I just erased what's there, but you can always pause and go back and look at it. Okay, our last paragraph. It says, in response to skeletal muscle injury, quiescent myosatellite cells are activated to become myoblasts. Okay. Researchers investigated how the extracellular microenvironment might regulate the expression of myoblast differentiation. Okay. So, what they're saying here is that the researchers are trying to see. So you want to figure out what the researchers are trying to study. So our IV here is going to be the extracellular microenvironment. So the environment that the cells are placed in, what the researchers are looking at is that's our IV. Our DV, remember when you're reading like experiments and graphs, you want to kind of figure out what is the IV, what is the DV, what are the researchers changing, and what are they measuring. Our DV is the expression of myoblast differentiation factors. Okay. So they said they're using urinary bladder mucosa. It has collagens and growth factors in it. So mice were given a skeletal muscle injury. So this causes the myosatellite cells to become myoblasts. Okay. And then they were either given the UBM, so our levels of IV are UBM, or just sham treatment, which is just control. Okay. So sham is just a control treatment on the MCAT. So we wanted to see how the myoblast differentiation factors, which we talked about earlier, MyoD1 and myogenin, how their expression differed based on what environment the cells were put into. So the cells were either put into a urinary bladder mucosa environment, or they were given a sham treatment, which is just a control environment. And first, the cells had to be activated to become the myoblasts through the skeletal muscle injury. And then after that injury happened, since they were now myoblasts, you can see now how the environment of the myoblasts affects their differentiation factors that they express. Okay? So now we have two graphs here about MyoD1 and myogenin expression. And what's really helpful with these graphs is to look at the caption here, because a lot of times they'll like simply tell you what the graph is showing. So right here, and always on the MCAT you want to look for stars. You want to look for stars because these will tell you if differences are significant on the graph. Okay. So here is our star right here. That tells us that at day 28, the expression of MyoD1, one of the myoblast differentiation factors, was higher in the cells exposed to the UBM environment as compared to the sham treatment. Okay, let's go to our next graph. Okay, now we have a myogenin expression graph. And you can see at day 28 in the UBM environment, there is also higher expression of myogenin, so higher myoblast differentiation into skeletal muscle. Okay, just like here, myo-D1. More myo-D1 expression is going to lead to more skeletal muscle. So the UBM tends to favor skeletal muscle through differentiation of the myoblasts. Okay, now let's finally tackle the questions. Okay, myosatellite stem cells can most likely be classified as what type of stem cell and is belonging to which germ layer? Okay, so this is a good thing that we can probably use Google to look up a good diagram to understand this concept. So what are totipotent? What are multipotent cells? Okay. So I like this diagram right here. So here we have a totipotent cell. So what is a totipotent cell? A totipotent cell can become any embryonic structure or extraembryonic structure, including the placenta, or the chorion, or the amnion. So any extraembryonic structure or any embryonic structure. So basically totipotent cells can do everything. They become an embryonic structure or an extra embryonic. Okay. A pluripotent cell can become any cell within any of the germ layers of the embryo. So when you have an embryo, it's going to have 3 germ layers: the endoderm, the mesoderm, and the ectoderm. These germ layers can give rise to different parts of the body. So if you have a pluripotent cell, it can become either the endoderm, mesoderm, or ectoderm. It can do any of those. We then have a multipotent stem cell. A multipotent stem cell can divide into any cell of one germ layer. So you could have a multipotent endoderm cell, which can divide into any endoderm cell. You can have a multipotent mesoderm cell, which can divide into any mesoderm cell. And you could have a multipotent ectodermal cell, which can divide into any cell of the ectoderm line. Okay? So main takeaways: totipotent cells can make up any embryonic structure or extraembryonic, such as the chorion, amnion, whereas the pluripotent stem cells can divide into any cell of any germ layer, and the multipotent cells can divide into any cell within one germ layer. All right, let's go back here. So they're asking us what type of stem cell can it be? Well, we know these myosatellite stem cells are going to become myoblasts. That is pretty specific, right? They're becoming into muscle cells, myoblasts, towards skeletal muscle cells. So this seems like within one germ layer. So we're looking at one germ layer. What did we say is one germ layer? What type of cells can differentiate into any cell of one germ layer? Well, we said that was the multipotent. We said that was the multipotent cells. Okay, and what germ layer are the muscle cells in? That is a good question. Let's see. Let's look up what germ layers examples. All right. This is a good chart for you guys to know, because on the MCAT they'll test you where each part of the body comes from. Like which germ layer? Okay. So where does it say mesoderm? Where are muscle cells? Okay. Oh, it says here the muscular system is in the mesoderm. All right. So what you want to know is the mesoderm is also known a mnemonic or a helpful learning technique. The mesoderm is also called the movoderm. So stuff that moves always comes from the movoderm. So, for example, muscles, bones, the musculoskeletal system, the circulatory system. Remember, moves stuff throughout your body via blood, and the adrenal cortex, which release steroids. These all come from the movoderm, aka also known as the mesoderm. The endoderm gives rise to, for example, the liver, the pancreas, and the epithelial lining of the lungs and digestive tract. And the ectoderm is kind of called the atractoderm. The atractoderm. So things people are attracted to, which is skin, hair, nails, for example, and also your nervous system. So those are the big parts of each germ layer. So let's go now to and also the kidney is also in the mesoderm. So that's something to pay attention to. Let's go back to the question. So we know it is a multipotent mesodermal cell. Good. Next question. Myopathy is associated with limited skeletal muscle development. Which of the following a developing organism is most likely to cause myopathy? So remember when we were talking about the factors that influence the skeletal muscle differentiation? Remember that complex, that protein complex that binds to the promoter genes and affects the skeletal muscle genes, so therefore it affects differentiation? That is what this question is testing. So what causes myopathy? So whatever is limiting the expression of the skeletal muscle genes is going to cause myopathy, right? So let's just quickly draw it out. We have the MyoD1. We have the E12. And we have the protein X. Okay, and they bind to the E-box promoter. So the E-box promoter DNA sequences, and it controls downstream the skeletal muscle differentiation. Okay. So what factors are going to affect this? So if we add UBM, here we saw UBM increases MyoD1 expression and increases myogenin expression. So these are factors that commit the myoblasts to skeletal muscle lineages. So it's going to increase skeletal muscle. So we can eliminate that answer because we know it's going to cause more skeletal muscle, right? We have more MyoD1 here. It's going to bind to the E box. It's going to increase the skeletal muscle. Okay. So let's draw. We know this is going to increase skeletal muscle, so that's not right. ID inhibition. What does ID do? Well, we said up here that ID inhibits the DNA binding proteins. So this complex here cannot bind to the E box, and it cannot stimulate its transcription of the skeletal muscle genes. So ID inhibits this. Well, if you inhibit the ID, then the ID can't inhibit this protein complex. It can't inhibit the binding to the DNA sequence. So the binding to the DNA sequence is going to increase, right? Because you have less of this inhibition by ID, because ID itself is getting inhibited. So you're going to increase the amount of skeletal muscle, okay? Okay, now you have MyoR expression. What does MyoR do? MyoR binds to and activates the E-box binding domain of protein X. So protein X can no longer bind to the DNA sequence because of MyoR. So if MyoR inhibits this binding to the DNA sequences, we're going to have less skeletal muscle differentiation. We're going to have less skeletal muscle. So this seems like the correct answer because myopathy is less skeletal muscle, right? So limited skeletal muscle development. So this seems like the right answer. But let's look at D, P21 clip expression. P21 clips upregulates the expression of MyoD1. More MyoD1, we're going to bind, this complex is going to bind to the E box. It's going to stimulate the transcription of skeletal muscle genes. So this will increase the skeletal muscle. Therefore, our correct answer is C. Okay? Erase. Next question. In myoblasts, MyoD1 most likely causes. All right. MyoD1 causes differentiation, right? Because these are regulatory factors. They control the skeletal muscle differentiation. Okay. So all the answer choices include UBM. So what does UBM do? UBM causes more MyoD1 expression, right? More myogenin expression. So, and we know that MyoD1 and myogenin control skeletal muscle differentiation, right? So what we know is there's going to be increased differentiation. So they commit myoblasts to skeletal muscle lineage and they induce cell cycle arrest. So we're going to have less mitosis. We're not going to have increased mitosis. The meiotic activity is only for germline cells, so only for like sperm and egg cells. So this is not a reproductive cell, so this is not right. So we can eliminate C and D. A is apoptosis. It does not cause apoptosis of the cell, it causes differentiation. Apoptosis is regulated cell death. The cell is not dying, it's just differentiating, so it's B, right? When you're differentiating, you're not dividing as much. So your mitotic activity is going to decrease, not increase. So 3 will be B. Which amino acid is least likely to be found in the protein X binding site of MyoR? So what is MyoR? MyoR binds to and activates the E-box binding domain of protein X. The E-box binding domain of protein X has positive residues in it. So the protein X binding site, this is myo-R's. So myo-R is going to have amino acids that bind to the amino acids of protein X. And it tells us that it binds to the E-box binding domain of protein X, which are positive charges. So remember, positive and negative attract. So for myo-R to bind to the E-box binding domain, which we know has positive residues in it, myo-R must have negative residues, right? Because positive and negative attract. So these are negative residues. Let's run X through that. Negative residues here. Okay. So if we had a positive residue in myo-R, that's going to repel the protein X E-box binding domain, right? So we don't want positive residues in myo-R. So that's going to be the one that's least likely to be found. So which amino acids are positive? Well, let's look up what the amino acids are so we can have a good idea. So our positive amino acids are going to be arginine, histidine, and lysine. Our negative amino acids are aspartic acid and glutamic acid. So we do have arginine as a choice here. So we don't want a positive amino acid in the myo-R site that binds to protein X. So we do not want a positive amino acid in the protein X binding site of myo-R, because that protein X has positive residues. So if the myo-R has positive residues, they will repel. So let's write that out. The positive residues here are going to repel the positive residues of the protein X. So in the site that binds protein X, you don't want positive residues because protein X itself has positive residues. So you do not want arginine there. Okay, next question. Last question, guys. Thank you so much for sticking around. According to the passage, myogenic expression is responsible for what feature of skeletal muscle? Well, we talked about here that myogenin is required for the fusion of myogenic precursor cells to either new or existing fibers. So it causes the precursor cells to fuse together to form multinucleated fibers. Multinucleated skeletal fiber muscle. So this is your skeletal muscle fiber. You can see we have one long cell with tons of nuclei in it. That comes from many cells fusing together. So each of the nuclei are now combined because the cells have fused together. So it forms a multinucleated fiber, the myogenin, right? The fusion of the precursor cells. So their nuclei now are all together, like all put in one cell now. One long fiber. So that is what is responsible for multinucleation. Presence of striations, that is caused by the sarcomere, which is the actin and myosin filaments. That's not related. The ability to contract voluntarily, that is from the nervous system. And their lack of branching, that is not due to the myogenin expression. So that's the end of the video, guys. So let's make sure we got everything correct. Let's erase here. And as you can see, we got everything correct, guys. Great job today. And make sure to check out our MCAT Max website, the social media. And we'll be offering free group tutoring and free individual tutoring for everyone. So be sure to check all that stuff out and let us know if you have any questions. Thank you so much, and I hope you have a good day.